Better? Like this? Okay. Yeah, I'm going to present some results from the cooperation we have in the University of Murcia, where I come from, southeast of Spain, and the lab of Mariano Higes in Marchamalo, Spain. So, I already said what is going to talk about. The pathogen influence on the genetic diversity is important to know, and we have seen already in the talks of we have this morning that keeping and maintaining the genetic diversity is quite important because it allows the populations to cope with different environmental, climate, and also pathogenic perturbations, as I say here. A reduction of the genetic diversity may lead or may cause a reduction also of the unemployment of the population, because some ecotypes may be lost, some important local adaptation may be lost, and when this event is happening, then it can be a loss of alleles, and some of these alleles could be related to the pathogen tolerance. So there is a debate about how the human management through the so-called domestication of the honeybee may reduce the population effective size, but also decrease the genetic diversity. But as we know already from the talk of Roberto, honeybees are not fully domesticated because the mating system, the mating is not controlled by beekeepers. The queen goes out from the colony, flies, and then makes different number of drones. In the Iberian Peninsula, between 8 and 24 drones. And these drones sometimes are not selected. So that's why it's influencing on the genetic diversity of the colonies. We know that there has been a lot of information on the newspapers, and this, some of you may know, this is the number of August of the journal Time. And there is a lot of information about how many colonies are lost and where is this loss being produced. And we know that in Europe there are different rates of colony losses, but also we know that there are a high intensity of colony losses in the US. In fact, in the US this phenomenon is known as colony collapse disorder, and is characterized by a different sort of symptoms. I'm not going to explain them because I think that there are several talks more focused on this symptom. But what is important is that several factors have been analyzed and discussed about this loss of colonies, and among them, one of the factors that has been evoked as a very important relation with this is the presence of different parasites and pathogens. So I'm going to talk about two of them, mainly Varroa destructor. I think you may know this mite. It came originally from Asia. It's causing varrosis, and in Spain it was detected at the 80s, and in that period, in that decade, it produced the loss of around 40% of the colonies. We also know that there is another pathogen that is related to these colony losses that is called Nosema. Nosema, there are two different species, Nosema apis, Nosema serrana. Nosema apis was already traditionally infected at these Mellifera colonies, and Nosema serrana was recently discovered, but now we know that it was spread before, and now it's worldwide distributed. I must say that Nosema serrana is a problem, and is related to the massive loss of colonies, especially in temperate climate countries as Spain. So it's interesting to know what's happening with the relationship between host and parasites, and genetic variation can influence the host susceptibility to pathogens. And the Iberian Peninsula provides a very interesting scenario to study these. Why? Because there has been a lot of studies about the Iberian Peninsula, and we know that there are two different evolution that came in contact in this Iberian Peninsula. It's not something typical of honeybees, it's something that we also can see in other insects and also in plants. So there are some studies done with mitochondrial DNA analysis, and we could see that this distribution of the different lineages. At the north we have this European lineage, and at the south we have this African lineage. So this is quite interesting, because we may wonder what is happening with these two different lineages. Are they susceptible in the same way to the pathogens, to the parasites, because they have different genetic background? So we did a study, and we tried to correlate the different evolutionary lineage with the presence of this the parasite, of the mite parasite, and also with the Nosema pathogen. So we determined the prevalence of honeybees in two samples from 2006 and 2010. In 2006, we found the Nosema apis was found in a significant higher frequency in this M that are located at the north of Spain than in the African lineage. This may be related with the climate, because at the north of Spain it's rainy, it's more wet climate, and then the bees are spending more time within the colony. In 2010, what we found out was that Nosema serrana increased the presence quite a lot in both types of honeybees, both in the European related and also in the African. In relation to the mite, we found no difference, no significant correlation between the evolutionary lineages and the presence of the mite. So once we knew that, we wonder what's happening then with the genetic diversity. So is being treated by the presence of these pathogens makes something in the host-parasite relationship. So we analyzed the genetic diversity through other markers, nuclear markers, called microcerealize in these two samples, and we analyzed the correlation of the genetic diversity with the prevalence of Varroa destructor and Nosema subspecies. We also wonder what's happening with these markers. Could we see some differences in allele frequencies that could be related with some loci related at the same time to the selection? So since the honeybee genome is a full sequence and some maybe annotated, then we try to look also for this. So we use the same sample as I showed you before. You can see here the different places where we took some samples. In fact, we took about 230 samples, and then we detected Varroa just by direct visualization, and also we analyzed the presence of Nosema by molecular techniques, one of them called PCR. So the results we can see about the genetic diversity in these two different samples are present here. We are able to see different parameters, but the one I would like to focus is the genetic diversity. This number, there are here and here, and you can see that they are not different. So the genetic diversity was more or less the same. Furthermore, we have information about a previous sampling made in 2000, and again, the genetic diversity was very similar, was not significantly different. So we can say that the genetic diversity, at least in the Iberian honeybees, maintained for several reasons I will try to explain later. But what happened with the presence of the different pathogens and parasites? First of all, at least in this sampling, we found out a reduction of the presence of the Varroa mite from 30% to 19%. But in relation to Nosema, we found out higher prevalence, higher frequency of infected colonies. And in fact, if we look about the distribution of the two subspecies, we found a higher frequency of Nosema serrana compared to in 2010 compared to 2006. So, since we didn't find a difference, at least in these two sampling periods, we grouped our samples into different ways. We grouped the parasitized colonies, that is, all of them that have or either Nosema or Varroa, and we compare the genetic diversity with those healthy colonies without the presence of the parasites. And we found out a slightly higher level of genetic diversity. But of course we have to test if this was significantly different, and it was not. I mean, we are talking about 0.02, but, well, we should maybe discuss a little bit later. So we then look at the frequency and the distribution of different alleles, and we saw that in these two groups, there was a difference of the presence of alleles, and also there was a difference in the frequency of some of the alleles. In fact, we were looking for the statistical genetic parameter and found out that one locus, that is called B124, was a candidate for a positive to external selection, because this value was higher than in the other ones. So we look about which is, how is this, where is located this locus? Is it related to some immune genes or something like that? So then we look for it, and when we find out that this locus is located on the chromosome 13, the Apis mellifera, and is associated with a gene that is called BPR7, and this gene is known that it goes for the immunoglobulin proteins, so they are related to the immune system. So with these results, we conclude that despite the observed variation of Varroa and the Nosema prevalence, the genetic diversity of the population has not changed over the last 12 years. We can explain this in two different ways. One of them is the uncontrolled breeding of queen bees with drones from different colonies. In Spain, there is no tradition of queen rearing, so the beekeepers really don't care about queens and about how they mate. They just leave the queens, and then the queens fly, and then they mate. But also, there is some influence of anthropogenic factors, because in Spain, the beekeepers do some transhumance. That means that they took the colonies and they move the colonies from one place to the other, not like in USA that they cross different timetables. But for example, from the north, they take the samples to the south of Spain, and the other way around, just looking for bloom. And also there is a tradition in Spain of swarm catching. The Iberian honeybee used to swarm, has a high tendency to swarm, and the way of increasing the number of colonies of the beekeepers is just to catch the swarms. They are looking for swarms, and then they introduce the swarms to the beehives. So we have found a slightly decrease of, although not significant, in colonies with the presence of the studied pathogenic elements. So this is consistent with the theory stating that the populations with lower genetic diversity suffer higher prevalence of parasites and pathogens. So this has been studied not only in honeybees but also in bumblebees. There are some evidence that those colonies with lower genetic diversity are more prone to suffer of pathogen impacts. So one of our last conclusions is that these microcellular locus appear to be subject to the selective forces, so that selection is active in these colonies, but only in those colonies that have Varroa. So since Varroa is in Spain for almost 40 years, it looks at some selection, some sort of selection is taking place there, and there's just want to remind you that the next, I will take my chance, the next European Congress will be called APIMONDIA, where I came from, next year in September 2014. So I would like very much to see you, all of you there. So thank you for your attention.
Thank you very much also for the information about the European conference. Are there questions from the audience? I see this correctly. You stated that there are colonies that are varroa free in Spain. They have an infection rate of 90% and something like this. So how do you define a colony that's varroa free?
In this study, what we did is just we took some workers from the inner frames of the colonies, and then we put methanol, and then we test for the pollen of the mites. So if we see at least one mite, then we label this colony as parasites.
And how big was the sample you took?
Sorry?
How big was the honeybee sample you took? How many bees did you test?
Oh, I took around 13 colony for the inner frames, where the blue bees and...
Further questions? No? Then thank you very much. The next speaker is Goblirsch, application of honeybee cell culture in biotechnology and agriculture.
Is that better? Okay, thank you. So this is work done at the University of Minnesota. My advisor, Marla, Spivak, and Dr. Tim Kurtti. And to start with, I would like to say that many of the challenges to bee health are from intracellular, such things as viruses and nosema. You have to get inside of a host cell and infect that host cell and replicate. So this causes problems for us because a lot of our studies are done at the colony level or a whole organism level. But really these things are working on an individual cell level. So it'd be good to have a tool. Louder still. Oh, closer still. There, am I getting better? So going back, viruses, for example, need to infect inside of the cell to reproduce. So to better understand viruses, it'd be good to have a tool, a cell culture system to understand what's going on. And tissue culture really is taking a fragment of the whole organism, such as ovarian tissue, for example, or gut tissue, bringing that into a sterile environment. Here you see the flask, and allowing the cells from that tissue to adapt to the growth medium that you give them, which is a physiological relevant medium for those cells to survive. And over time, it's hoped that those cells adapt to life inside of that flask, and they begin to multiply and divide, and then you can take part of that material and split it into a daughter flask. So you start with a primary flask, that material grows out, then you can have to transfer some of that material to a daughter flask, and then continue that process again. Now, I put some time here. Starting with primary culture, it may take months before that material transfers to a subculture. And then again, the adaptation period may again be months from subculture to the next passage. Over time, though, those frequencies become, the interval between those frequencies become more frequent. So less time between subculture. So with insects, we have over 500 insect cell lines available, but very few from the Hymenoptera. Less than 10 cell lines have been established from bees, wasps, and ants. So it's long been thought that honeybees, for example, may be refractory to growth inside of the culture system. We don't have a honeybee cell line. There's been some recent work where transgenes have been used to maybe immortalize a cell line that way, inserting human oncogenes, for example, to try to develop the cell line through other means. So this is where my research has started. How can I establish a cell culture system to promote a long period of survival of a primary culture, which would then increase my chances of adaptation to a cell system. And what I did was the methods were pretty straightforward. I used embryos as my tissue of choice. Embryos have a lot of mitotic potential. So I would take an empty frame, insert that into a colony, allow the workers to clean up the cells in preparation for the queen to lay eggs, and then cage a queen on one side of that frame. And that's my time point 0. After 24 hours, I release her, we go back to the colony, another 48 to 24 hours later. So right almost at the end of when the eggs are starting to hatch, I pull that frame from the colony, and I bring it into the lab, and I collect my embryos. Those embryos are collected into little flasks. I have to sterilize them, do some bleach, some ethanol, and then I mash up the embryos, make them into little fragments. And I introduce those fragments into a culture medium, and then I watch to see what happens. Between 2009 and 2012, I set up 92 primary cultures. And I put that up there to say that not every culture turns into a cell line. There's a lot of trial and error, and with experience, most of these cultures that I set up, I was able to then, they had a longevity of about 6 months, which is pretty good. Previous reports say about 3 months. But the longer that period, that primary survival, the better my chances were that I could start that cell culture process. Get that line going. So again, 92 primaries. Out of that, I threw most of those away. Again, about half of them survived greater than 6 months. My low success rate also led to a long adaptation period. And what I'll say is that one of my 92 primaries shows this profile of establishment, of passage. So meaning, if you look at time point 0, is when I started the culture. And then each of those points is a passage or subculture. So on the x-axis, it's cultures in days. So we see 800 days, a little over 2 years. This is one primary that I established. If you look for the first year or so, the transfer history is pretty slow. I only subcultured that one primary about 5 times in a little over a year. And then there's this long lag period at about between 300 and 400 days. But then something happened. My cells were transformed, or they adapted to that artificial system. It's an artificial medium. It's not. It's removed from the whole organism. They're growing on a plastic substrate. They have a culture liquid that is approximate, but so something happened. I can't pinpoint exactly what happened, but the frequency of the cell culture then became bi-weekly. I can transfer material every other week. And this culture that I established started in July of 2011. And I've named it AME-711, Apis mellifera, July 2011. And this culture is still alive and it's still growing. I have to show that these cells are honeybee of origin. And to do this, I look at the chromosome numbers. 2N in honeybees is 32. When I prepared some chromosome spreads by blocking them with in metaphase arrest, and then count the number of chromosomes. For the most part, most of my cells had a typical diploid chromosome number. I also looked at a genetic marker, cytochrome C oxidase, which is variable across species. So the expression within honeybees should match what my cell line was, using primers that are specific for cytochrome oxidase within honeybees. So you see my lanes 2, 3, and 4 are honeybee origin. They're an adult bee, they are an embryo, and they are my cells. And then I use negative controls, patient's larvae, a bumblebee, and a human cell line, and some PCR controls. So when I amplify DNA from these different samples, I got a positive signal for what I would expect, Apis mellifera, adult abdomen or adult embryo and cells, and no signal for the other three. And I also sequenced those products and for the embryo and the cell culture, and they match as well as previously published sequences for cytochrome oxidase. And what my cells look like in culture, here's 4 panels. If we start with A, that is a tissue fragment. That's day 0. That's a part of an embryo that I've matched up that's floating in medium. After 24 hours or so, here is my tissue fragment, and outgrowing from that is very regular shape, I'll call them epithelial cells. And this is 24 hours after inoculation. Within weeks, my culture becomes very heterogeneous, lots of different cell types. Some of them single cell type formation, which I thought could be hemocyte. I'm not sure. My tissue fragment here begins to disintegrate, I guess, over time. And I guess as I move forward, this is my current cell culture right now. It's become mainly a fibroblast type cell system. So either way, I've selected for those fibroblasts because they're adherent in my passages and my media exchanges, or the other cells have died, but it has become dominant with the fibroblast types. So as I mentioned, the cell culture is still alive, and what this will allow, I believe, is we'll have homogeneous replicate cultures. Think of setting up a 96-well plate with different cells, and then being able to test, maybe toxicity testing, dosing, or also looking at some of the pathogens, the viruses, and then look at, examine infection, transmission, or the host response within a simplified environment that is outside of the whole organism, and outside the complications of the colony. These cells have been cryopreserved. They can be stored in liquid nitrogen, and I can recover them from liquid nitrogen. So this will then allow also to then distribute them to other labs. Another thing that will be useful is that it will be a platform for transgenesis. Think of being able to or by being able to use transgenic technology. And with that, I'd like to thank those that helped me on this project, Willie Munderloh and Dr. Kurtti's lab, and Fallon, and just a small University Award for the funding. Thank you.
Thank you for the talk. Are there questions from the audience? I'm not a specialist in this, but what is the reason that it's so much more difficult to get cell lines from insects than from other species?
I think the difficulty isn't so much in insects, but Hymenoptera in general has been a difficulty. And that we don't know why it has been a challenge. It's very easy to get a cell line from, I'll say, relatively easy to get a cell line from Dipter, for example. Hymenoptera has kind of eluded us.
But again, I think we don't know why it's so difficult, especially with Hymenoptera.
Right, we don't.
Did a question arise meanwhile? Yes, please come to the microphone.
Short question. There is a range of stem cell cultures, obviously tissue culture, try those use of the other process for culture, any
We do not get any stem cell factors in culture.
Okay. Thank you very much.
My topic is 3G would like to be related with loyalty yield and using G chip. Loyalty they develop in the case development decision between green and the woods. And the loyalty was also well known for its high effective function for human. Up to now, more than 90% loyalty was produced in China. There are about 3,000 tons every year was produced in China. And this work, I think, is contribute to the beekeeper in China and the scientists on loyalty producing technology, especially for the loyalty and production breeding. The representative breeding is general number one honeybee. And this breed, the production arrived more than 200 grams for 3 days. And many scientists also make research on a genetic base related to loyalty production trait, such as morphology and cytology, biochemical marks, and molecular marks also. For molecular marks, some people make micro-satellite. They find some satellite related to loyalty production trait. And Professor Lee also made use protein technology to screen some marks. And also some PhD-1 made screen DNA some fragment related to loyalty production. With the development of the molecular biology of honeybee, and especially for the genome sequence of honeybee was completed in 2006. So we can use a lot of that from the NCBI, and also we use new test gene sequence, and some protocol is related to royal jelly production genes to make new gene chips to screen this trait. Honeybee sample collector. We use 4 colonies with high royal production and 4 low production. And these high colonies and low colonies was used hybrid and inbreed method to produce these colonies. And also we determine their production of larger. We collect 30 head of nursing bee. When we take out royal jelly flame, and then the nursing bee, when they are feeding in the queen cells, we take this individual into liquid nitrogen immediately. And this is the production of these colonies were defined. You can see they are different significant. And this is the process. We extract RNA from these nursing bees and transfer into cDNA and then hybrid with the chip to get the different genes results. We found 369 different expressed genes. Among them, 168 down expressed and 201 up expressed between high and low color. We make bioinformatics analysis of these different expression genes, including PC, cluster analysis, and gene analysis, and pathway analysis also. This is the result. It's PC analysis. We can see from this picture that this growth this gene relate high production colony, and this area is low production colony. This result indicate that our samples they are different growth, high production growth and low production growth. This is from another angle to analyze PC. This result is the Caste analysis of these genes. These 368 genes, and the left side, 4 colonies, is low production, and the right side is high production. We also make process enrichment analysis of this gene, and also from molecular function analysis. This is the KGG analysis for this gene. Another work we do from these different genes. We want to verify which genes are really related to the royal jelly production. So we use we verify 46 genes. These genes travel significantly express genes between high and low flow. And 18 purely local gene in NCBI, and 8 most common gene associated with trade and other tribal genes. This is the primers of these genes for qRT-PCR. This is the result of the qRT-PCR result. There are 10 genes we found is really different between these two groups. We collect other new samples, anti samples, to verify this result. We found from 46 genes, we screen 10 genes, they are different. Yield and gene expression level, because they are quality. So we can make the lines between gene expression level and the larger production. We use 7 colonies. We can see the right side is the production, and the other lines is the gene expression level. From this result, we find 3 different genes. These different genes, we can analyze their correlative is significant. We use DPS software analyze. We can see the efficiency is high. This is the reversion analysis for one gene. The public research 2 gene. And this is signal research gene. This is X71 gene. So we get a conclusion. These 3 genes, up to now, you can say they are related to the larger production. Discussion. This study provides some gene related to the larger production, and we are going to continue to make research in this field to discover more molecular mechanisms of the larger production. And these molecular marks we think is also very useful for our breeding or larger production trade. For public research gene, we can have a hypothesis that maybe clones to recognize the breed lab. This is just a hypothesis. And some of my students continue to work with. We use RNA influence, and also qRT-PCR and other molecular technology. The second gene, Hex71, delayed the transmission from losing to foraging. We think this is also very important. Also, the nursing period of the breeding, high production breeding, is longer, the nursing period. So maybe it's related to this gene. So this is our procedure. More research in this field. Acknowledgement. I'd like to thank to the government from cultural ministry of China give our money to research in this field. And also Professor Chen, Professor Zhang from AIU give our help. Professor Dr. Sakti from USA to help us. And I also thank several of my students work very hard in this field. We get this result. Thank you for your attention.
Thank you. Some questions? As mostly I have to start. So if Rob Page is breeding his bees so much in a certain direction that they only collect pollen and nothing else, they will have a problem. Do you see things like this if you breed bees in the direction of producing a lot of royal jelly? Are there any deficits in those bees? Do those bees be weak in some concern?
In China,
Yes.
until now, we still find this phenomenon with some of scientists also made investigation of this but in China, and for royal production, the talent should be very strong, otherwise we cannot produce royal. So we also care about, now we don't care about the royal production, high. No, high is enough. The quality is important. The honeybee health is our next goal.
Thank you very much. Any question? So if not, then thank you very much for the one. And the next speaker is Roberto from Brazil, unrevealing the molecular mechanism determinants of Caste development, the honeybee apis. Could you please thanks to this organizers and to the speaker. It's a pleasure to be here to share some more findings on caste development and this. Some distinguished bees. One I feel that we receive a lot of attention during development, and the other as Page said before, I received different attention, and it's a kind of differential feeding. And this differential feeding during development
give rise to very different phenotype here. You can see here queens and workers. There are a lot of morphological differences between them, and we are focused now on this. For example, workers have here a kind of body press, and they have a larger brain, kind of larger brain, and queens, for example, have enormous and so on. And this, the way these two morphs originated in the development, the main question is sociobiology. Since before to 1941 or something like that, people were asking about the way these two castes originated from a single one young larvae. And the first systematic approach to this question was done by the group of Jara. He showed that a lot of food in some way give rise to a big-bodied female, and with the participation of some kind of hormone here. Then it was proved that that hormone was juvenile hormone. By application of juvenile hormone experiment done by Stalin in the 70s, something like that. And the first molecular approach on this issue was done by Severson in 1989. They got two sets of protein from developing there in wings and workers after doing in vitro preparation. And after this work, we have a lot of other different works on this issue. Done by J. Evans, Will, Miguel Corona, even Klaus Harfeller and his students. All trying to answer the question in a kind of systemic approach. Using the whole body. And the whole idea was this in this work. The differential feeding in some way would give rise to a very different hormonal environment. High levels of juvenile hormone in queens, low levels of juvenile hormone in workers, and in some way this environmental, environmental would give rise to two sets of gene expression, maybe a specific gene expression. And these genes, in some way, will give rise to very different environmental trajectories. By the time we started working with this issue, we have this picture here. High levels of neuron giving rise to this governing the expression of kind of physiometabolic genes. And these physiometabolic genes will be responsible for the incremental alteration, giving rise to the queen phenotype. On the other hand, low levels of juvenile hormone would be responsible for some kind of developmental genes, and this would be responsible for the development of character state alterations, and thus giving rise to the work phenotype. This was our equation by that time. And to answer the question, we carried out DNA microRNA hybridization using RNA sample from developing queens and work lab. And we got 240 differential species genes. Then we look for here, in the upstream region of these genes, we look for a cis element like this one here. Cis elements like this. This region, upstream region of these genes, trying to get clues on how these genes, 240 genes, could be regulated during development, and thus give rise to the different phenotypes. With this information, with 240 different genes and cis element, we elaborate these two complex networks here, mainly done by Alexander Gichino, friend of Goblirsch. We have this network here, characterizing queen phenotype, that is queen here, and this network here characterizing the worker phenotype. And it mainly allows us to identify something like this element here. Using these networks, we can identify the interaction of this element with this other element here, and conceive some experiment and test the real participation of this gene in the whole process. Using this network here, and we had by that time, we proposed this general model of caste differentiation in Mellifera. It was this simplified model, working model, in which we can see here a high amount of royal gem would promote low levels of high levels of tor activity here, but it's a kind of kinase. This tor activity in some way would give rise to high levels of juvenile hormone, and this would govern the development of the queen phenotype. On the other hand, the worker gem would promote low levels of tor activity with low levels of JH, and this governing the development of the worker phenotype. Interesting, because by that time we didn't know this exactly this link here. A work here published by José Luis Maestro and Javier in Barcelona using La Tierra Germana and Goblirsch. They, in 2009, proved that actually is tor activity sensing nutrient availability govern the synthesis of juvenile hormone. This picture here. Tor activity, high levels of nutrition, high levels of tor activity, and high level of JH production. And now we know that this is true also for Mellifera. And this link between tor activity and queen development simultaneously here proved to be true by the group of Luanda. You see the kind of down-regulation using RNA I for tor in prospective queens. So they beautiful workers like this one here. So put the general model proposed in 2007. And 2011, Kamakura showed this elegant result here, by which he used royal actin, a small protein from the royal gem, and he proved that this protein gives rise to its responsible in some way for giving rise to the queen phenotype. He also used the fly as a model, and he obtained a kind of royal fly there, beautiful, big, female, and so on. However, there is a kind of problem with this result for some part of the scientific community, because this result doesn't fit very well this model here. Maybe probably we have redundant pathway here, including the insulin pathway, tor pathway, one factor receptor pathway, and here's ligand, and royal actin. By that time, so it was clear that we have a kind of morphogenetic field here operating in the level of development that has given rise to different morphologies in queens and workers. If we look here, ovaries, brain, and hind legs would be a kind of morphogenic field. And we started to study this morphogenic field and how this originated in development. The first study was done actually by the group of Klaus Alfelder, who is here. Klaus Herfelder in '98 and his students. He showed that high levels of juvenile hormone in some way prevent cells in ovaries prevent from apoptosis, and it doesn't take place in workers, so where we have small ovaries and and so we started to work with the pollen collecting apparatus as a kind of second morphogenic field. And with Anna Durvalina, we using scanning microscopy and mineralization, we showed that the pollen press, this region here, hairless region with the big hair here, present in workers, is developed only in the presence of the pressure of UPX. In queens, we don't have this impression, and we have a very surface here, we don't have a polyethylene apparatus here. We are working on some functional study now in order to test the real participation of UPX in the products, but have nice results. And the third morphogenic field we are working on is the brain development. Since the workers need a lot of brain functions, they have the children larger brain, larger brains, and we started studying this development of this organ during larval period. And surprisingly, we found that queen's brain is larger than the worker's brain. You can see here the calyx, the whole brain is larger than the worker's brain in L5, for example. Using immune incorporation here in the cell during larval development, we showed that queen's brain have a lot of more proliferating cells than worker's brain do. And this takes place during the whole larval period. So, summarizing, we have this picture here. Queen's brain carries develop larger and are earlier than in worker's brain. We studied some genetic genes during the whole process. Using qPCR, we showed that at least 3 genes are differentially present between brains, queens and workers. Krupel, FAX, and Songkun. Then, using this in situ hybridization, we showed that SHOT, for example, is present in queens and is not present in workers. Using new organization, we showed that the same SHOT protein here, SHOT protein not expressed here in workers. And SHOT protein is a kind of cytoskeleton protein responsible in some way for structuring neural development in animals. And we have to, again, after this screening, we have to test the extent of SHOT participation in the process. This is, if we look here, L3 brains, they look pretty similar here. So we are trying to get molecular evidence on maybe the different, some kind of differentiation between these 2 brains in the L3. Before L4, that is the state we see morphological differentiation. So we are using RNA sequencing experiment. With this tool, we got 11,000 expressed genes in L3 brains, and 60 are differentially present genes in L3. So we have this distribution here. We are now working on some in silico analysis using prediction of gene regulation and how this could be responding to differential feeding during development, mainly in brain development. I have to say these people all participate in these boards, and this is Ruben and Feras. Thank you very much.
Thank you very much. There are questions. So when you pointed out the brain is developing faster or slower, what does it mean for the quality of the brain? Because the facilities of the brain in the development period, that's the thing that's important.
Yeah, actually we have here a very important issue. I said in the beginning that in adults, warriors have a relatively large brain than twins. However, this result here shows the opposite. So we have during the next developmental stage is metamorphosis, and we have the answer to this question. We are working on this event nowadays, and we found some interesting result from here, in which the warrior's brain shift the expression of some genes, immunogenic genes, and the morphological aspect of the brain during metamorphosis. This is our late L5 state. So we have the next year some result.
Thank you very much. Another question? If no, then we are going to the last talk. It's by Pilar. Thank you very much again. Genetic diversity and honeybee lethality.
Morning, everybody. Can you hear me? 3 seconds. Okay, that's fine. I'm here to present a talk. As you can see, I have a lot of co-authors. We also go under the name of Colors Working Group 4. This work that I'm going to present is really a work of all of these people. I think I even missed a few. A work where we try to study the impact of genetic diversity on colony mortality, and of course, being a Colors working group, this is related to the big losses of bees that we have seen in the past decade. So, where does honeybee genetic diversity come from? Well, the honeybee queen lays 7 to 25 times in open air. This is a way that the colony becomes more genetically diverse, simply by having all these males present with our offspring. As a result, you can find colonies like you can see on the picture here, with bees that are yellow, bees that are black in the same colony, originating from the same queen, having different fathers, they turn out looking different. This is something Rob mentioned in his talk. This is known for quite a long time, that bees have this genetic diversity, and it's much higher than what actually we expected if we go back 50 years in time. We saw 7 males, 20 is quite common actually. What does this mean for the colony fitness? Well, it's been shown in a number of studies that colony fitness actually seems to improve based on this multiple mating of the queens. There's lower susceptibility to diseases in bees, and also something like thermoregulation is better in bees, where you have a higher diversity of the offspring. And we don't know exactly how and why this happens,
but division of labor, as also mentioned by Raquel, is one of the mechanisms that we can think of to promote this higher colony fitness. But I'm not speaking about fitness here, I'm speaking about vitality. This is two very different terms. Fitness is quick to define, it's a number of offspring that you bring into the next generation. Vitality is something very different. Vitality, we have tried to define it. It's a capacity of a colony to cope with environmental stressors. So a colony's vitality can be, or is constantly tested by environment. It can be climatic variations, it can be lack of forage, it can be pests and parasites. All of these things will try to, some of them will try to impact on the bee health. Some of them are just happening by changing weather and season. It takes a long time for adapting in an evolutionary sense to set in for the bees to be able to cope with these local conditions, and this is an important factor in colony vitality. So, as I said, lack of forage can be a stressor. This is a picture from home, from Denmark, where you have green fields everywhere, but there's no flowers around for the bees. This is something that we humans have created in the past 100 years, very effective treatment of weeds, and this impacts for sure on the bee health, but the bees, I think, have not yet had a chance to evolve weeds to deal with this. But even before we had these changes in the environment created by humans, we already had different environments in Europe. And local bees, that's been shown quite nicely in a study from France, local bees are actually adapted to the conditions that they're in, and they start to produce more brood. When the flowering is starting in the middle of the summer, the brood production goes down, and at the end of the summer it goes up again when there's a new flowering setting in. So bees have adapted to these local conditions over a long time. There's also some stressors. Pilar mentioned one, Varroa as a new stressor. Very few bees have adapted to it, and therefore the losses are very frequent due to Varroa mites. Also because we don't allow the bees to adapt to it, because we go out and treat our colonies almost every year to keep the Varroa load low in the colony. Beekeepers are breeding bees, especially the queen breeders are, and it is in the past it was very simple. We would produce queens from the best colonies. This has been improved on in the last years. People are using isolated mating stations, either on islands or in valleys, and some people are even using artificial insemination to also be able to select the male side of your breeding scheme. However, this is also having some consequences. So the mating control has been proven very effective to reduce swarming, reduce aggressive behavior, and to improve honey production in colonies. So some of these mating stations that I know of, they have 15 colonies of sister queens producing drones for mating, and the nice thing for the breeders is that these drones are then either brothers or cousins, so you have a minimal genetic diversity. You can be pretty sure of the offspring of the colonies that come from queen and mate with these drones. So you get these gentle bees that don't swarm and produce a lot of honey. And particularly in Denmark, we have more than 20 islands. Denmark has more than 20 islands, but we have 20 islands that we use for mating to make sure that the bees mate with the males we want to mate with. In other parts of the world, they use mountain planets to achieve the same. But the result is a genetic drift, because you actually reduce the number of genes that pass into the next generation by selecting both on the queen side and on the male side. So how does that affect vitality? Well, for sure, this artificial selection, any artificial selection, will reduce genetic diversity. You throw away the bees that you don't want to have. So this is known for a long time. And beekeepers know that this has negative consequences. You lose diversity, you lose alleles in the sex locus. So to counteract that, some breeders produce hybrids between different subspecies. This can work very well. However, often these bees that are brought in are not locally adapted. So you get high honey yields, you get gentle bees, you get bees that don't swarm. But what we do find is that the vitality of the colony can be negatively impacted. So not all genetic diversity is beneficial. It's not just a matter of increasing your genetic diversity to the highest possible. You actually want to have locally adapted genes in your bees. So how can you breed for vitality? Is it even possible to breed for vitality? Well, it's not a very easy task. You need large gene pools to avoid this genetic drift. This mating and isolation is not very well suited. I really think you need to use locally adapted bees. You need, maybe for some of the breeds that have already become pretty rare in Europe, to use beta population structure to create transects for bees to move between isolated islands of occurrence. And some of the things that you really need to focus on and lack of swarming. Now, I'm already at my last slide, so I'm a bit ahead of time, I think. But this is, I would start by saying this is in the cooperation of a lot of people. We actually set up an experiment that we are basically publishing. We shifted bees around between 16 labs all over Europe. And the not so surprising fact, but it's very nice to actually have shown it, is that the local bees are doing much better everywhere. So, Italian bees do better in Italy, Greek bees do better in Greek, Macedonian bees do better in Macedonia, Danish bees do better in Denmark, German bees do best in Germany. It is really true that local is better, as I'm going to say in South Africa. You should not try to bring bees from the other end of the world. You should really in the future, if we want to find these lost colonies, I think we are many from bees. You cannot maintain this population on a long-term basis. But of course, this is something that needs to be developed, because we don't know how many bees must be transferred from Norway, from Netherlands to Denmark. We have no clue about these things. So this is something that is really a task for the next years to find out how to keep our bees alive on a long-term basis. I hope it satisfies your question. We talk later. Yeah, not a real satisfied.
So when you mentioned we have problems with diversity of bees in Europe, how about the United States?
Well, some indication from this colony losses in America is that the breeding in America goes back to, I think, 77 bee breeders. It's a very low genetic diversity. None of the bees in America are locally adapted because they have produced European bees. There's no point in being locally adapted in America because you put up a lorry across the continent every few months. It is a huge task in America to be that will be able to sustain, tolerate this type of beekeeping. I think this is part of the reason why it was because.
Thank you. That's a question waiting.
Pim Ras from the Netherlands. I think you said that to bring the Danish local bees on a desired size, population size, you want to import bees from the Netherlands, France, Norway, but that's not local anymore.
That's not local anymore.
That's fairly contradictory.
I know that we have done quite a lot of analysis on this question. Of course, the genes that are in Mellifera bees everywhere in the old part of Europe, that Mellifera heard, are not exactly the same. But the evolutionary history of these genes go back to the end of the Ice Age. And of course, most of the genes have a common history. But of course, you're right, it's really a predicament. How to introduce few enough genes, not to change the local populations, but have enough genes not to lose the vitality of the bees. This is a challenge, and I don't know exactly how we're going to solve it, but I can see if we don't, we lose our bees.
You were also suggesting that to keep local populations everywhere, but they are usually very small, isn't it?
They are very small.
It's a huge problem to solve.
It's an almost endless problem to solve, but if we don't start, we will finish.
Robert?
I would just like to address your comments about North American bees. First of all, on the slides that I didn't show, two of the slides that I didn't show, that I flipped through, showed genetic variation for at least 40 different traits that we looked at in North American bees. Absolutely everything we looked at, we have found genetic variation for, with the exception of Varroa. I mean, my own personal studies, where I tried to do a selection experiment for Varroa resistance on a large scale, it failed. So that's the other point. The only thing we ever failed to select for was Varroa resistance. We selected for many other different kinds of traits. So, my mother used to say that the proof of the pudding was in the taste. So when you look at the success of the breeding programs, they have been very successful, so there had to have been substantial genetic variation. So I would argue that that assumption is not correct, that there is in fact significant genetic variation. The other is the idea of adapted populations. In the early 1990s, I did a study of genetic markers in feral populations across California. We identified 220 different wild colony nests that were distributed between Southern California and Northern California and East and West. And what we found is in the feral population, there were kinds of markers which had been locally adapted over time. We can see the evidence of the selection. And they were different from the commercial bees that were close by. It had different markers. There were no clines in the commercial bees. They were just mixed up. Varroa came in the early 90s. Within 5 years, wiped out more than 85% of all the feral population. And every colony that we surveyed subsequently was either dead or heavily infested. When we went back 5 years later and looked at those same nest sites again, bees that we invaded them all looked like the commercial bees. They did not look any longer like the feral bees. So feral bees, or the honeybee was imported into California in the 1850s. So by the time we did our study in the early 1990s, we did have an locally adapted feral population that was eliminated in 5 years by Varroa. So that was our story.
I'm from England, and I advocate very much what you're saying about breeding our own local queens and our own local bees, because I think they are best for where we live. I imported a buckfast one once. I bought one, and I lived in the middle of England, and they killed us straight away. They didn't like them. Look, what I really wanted to ask you was about the Denmark bees, because from what I've read, you do use a lot of buckfast queens. And is this different to what you're doing in your work? Are you using buckfast queens or buckfast strain, or are you using them on a local one? And how is it different from Apis mellifera and mellifera?
I'm as guilty as everybody. We have all kinds of bees in my institute. We also have some of the local bees, original local bees, but we have bees of various subspecies, and it's not good. I really think we pay more attention to the local bees in the future, and not just economic production or gentleness. We need to focus on conserving what is there as soon as we can. Time is running out, and I think it's at least in my country, it's a benefit. We are not foreign colonies on one island, and I don't think that's a long time.
So your group not advocate using the buttress bees that a lot of your country
does? I think my job is that I can't really make any comments on that. People can do what they want to do, but I can try to educate them to do what is right.
Thank you.
I think that it's a nice word to be the last word. Is there any urgent question? So if not, thank you very much to all the speakers. Thank you very much for listening. And we have a second session in the afternoon in the Dining Hall with similar topics as we have covered now.